HPX2008 and HVTN 705. We also have other names, Imbokodo, HVTN 706, also Mosaico. That's sometimes easier for people to worry about. And we'll share you the current thinking on our plans for correlates and sieve analyses in this program. So all of this work is part of a very extensive public-private partnership effort with many people who've been involved in this program over its last 12 years. And our goal is not to work on a strain-specific or clade-specific vaccine, but a global prophylactic vaccine that'll be relevant for the globally diverse strains of HIV. And that's based on mosaic antigen designs developed by Betty Korber. And those are delivered through the use of a heterologous vaccine regimen with the mosaic antigens for GAG, POL, and ONV encoded by Ad26, as well as soluble trimeric GP140 envelope proteins. So in consideration of evaluating efficacy, we're obviously worried about clade diversity, but also making sure that vaccines are relevant against multiple modes of transmission and also valuable in the context where PrEP is widely used. So currently for the program, there's two complementary efficacy studies ongoing. HVTN 705 is Imbokodo, 706 is Mosaico. Imbokodo is in 5 countries in Southern Africa, whereas Mosaico is throughout the Americas and Europe. Clade C is the predominant viruses circulating in the Imbokodo study, whereas in Mosaic it'll be Clade B. We'll evaluate heterosexual young women, age 18 to 35, compared to MSM and transgender individuals, 18 up to 60 years old. And the modes of transmission between these two are also different. It'll be primarily intravaginal transmission versus intrarectal transmission. So combined between these two studies, though, there is a good coverage for both the major clades that are circulating globally, as well as the major populations and routes of transmission that need to be addressed. Mosaico, this is a quick overview of the study design. So 1-to-1 randomization. Participants get 2 vaccinations, either with the tetravalent Ad26 mosaic, followed by 2 more vaccinations at months 6 and 12 with the Ad26 mosaics combined and clade C GP140. They're followed up to month 24, and then in a stage 2 analysis until month 36. Mosaico has very much the same design. The only difference is that in the 3rd and 4th vaccinations, there's also the inclusion of the mosaic GP140 component. This was shown to expand the clade B and non-clade C immune responses while not interfering with the clade C responses. So we think this will be a better antigen combination for a globally useful vaccine. So there's also different types of immunobridging. So these 2 studies will evaluate if the vaccines are efficacious in specific populations, but in order to support a broad evidence package for the use of these vaccines, we need to conduct studies that'll bridge between these 2 regimens to show that what happens with the regimen in Mosaico is also relevant for the regimen used in the Imbokodo study. We need to bridge between populations to show that protection that would be relevant occur in young women in Southern Africa is also relevant for young women in other parts of the world. Also to bridge between clades to show that if we can protect with these regimens from clade B and Mosaico, that other MSM and transgender individual populations could also be protected in other parts of the world. We're not going to be able to bridge formally between modes of transmission, but given that the modes of transmission that are being studied are those that are generally the higher risk of infection per exposure, we think that it would be reasonable to argue that meeting a high bar for transmission risk would also protect against other modes of transmission. So currently in terms of what's going on in the Imbokodo study immunogenicity evaluations, there's a pilot study to characterize the, well, there's four objectives of it. This is occurring right now while the follow-up stage of the study is still ongoing. We're going to characterize the immunogenicity of the vaccine regimen in the study population. So we've compared we've characterized the immunogenicity of this vaccine regimen in general, but we need to understand how it actually behaves in this population. We want to understand the temporal relationships between the immune responses, factors that could influence immune responses at baseline, then post-third, post-fourth vaccination, and then 1 year after the vaccination to see how those responses relate to one another. That'll be informative for any time-dependent correlates of protection analyses that could be conducted later on. We also need to develop best possible predictors of immune responses, such as you might consider Ad26 neutralizing antibody as an example of a potential baseline predictor, or age or body mass index or things like this. And also based on these pilot study results, we'll be able to down select the unique markers that'll be included in the first tier of case control endpoints for the correlates of protection analyses. So these are the currently planned pilot immunogenicity evaluations, looking at binding antibodies to the vaccine matched envelopes, BAMA antigens against sets of clade C envelopes, looking at IgG3, IgA, and IgG responses, phagocytosis responses, cytotoxicity responses in the infected cell assays, the CD4 and CD8 functionality, as well as T cell breadth evaluations by ELISpot. And ELISpot will also look at the responses to the PTE peptides that were used to help design the mosaic antigens. And we'll also evaluate the pre-existing vector immunity. In parallel, we're also initiating the sieve analysis for infections that occur throughout the first stage of the study. So we'll sequence all the viral genes that are encoded in the vaccine, so GAG, POL, and ONV. This will help characterize differences between the virus sequences that occur in the two study arms, so we can see if there's differences in the number of founding viruses, distance to the vaccines, if there's specific sieve effects that are elicited by the vaccine, and this will help elucidate the mechanisms of an imperfect vaccine regimen. And that will also then guide future iterations and more specific hypotheses that can be tested in immune correlates analyses. So, and then thinking further along, the Mosaico study is still enrolling, but eventually that study will need to also conduct immune and correlates analyses, and we want to do that having learned as much as we can from Imbokodo at the time. So everything that we've learned from Imbokodo, or that we're planning on for Imbokodo, is based on what we've learned through the previous clinical studies, most through RV144, the 505 studies, the STEP studies, as well as the NHP challenge studies. So all of the learnings that we've taken from these will hopefully give us the best possible chance to identify the immune markers that can be associated with risks of infection, and help define exactly when and where and how a vaccine like this could be used. So also when we do have results from Imbokodo, this will then allow a specific set of correlates hypotheses to be defined and tested in Mosaico. So beyond the two efficacy studies, we will also want to explore if it's possible to extrapolate from the clades against HIV is evaluated to other clades of HIV. Where our hypothesis is if efficacy can be demonstrated and we see similar levels of immune responses across clades, then we'd like to infer that vaccine could also prevent infection of those diverse strains of HIV. And that needs to be supported by the identification of strong immunologic markers that are associated with vaccine efficacy. So if Imbokodo would demonstrate efficacy in young women, then we'd be able to establish, hopefully, that other clades could also be protected from vaginal transmission, and correspondingly, Mosaico would also establish that efficacy for clade B and other clade protection could also be inferred for intrarectal transmission. So I just stop by thanking all our partners, many of our longstanding partners, our Janssen compound development team, clinical team, and our leadership, the Imbokodo, all of the Imbokodo sites, Mosaico teams, and thank you for your attention.